So I'm very, very happy to be with all you here. I mean, this is very important for us as Blue Mission, this first webinar. I mean, and I'm very, very happy that we have ClimaRest with this fantastic team over here to explain, I mean, their demo sites, these Atlantic and Arctic Lighthouse weekly hour will be very much focused on solutions. As you know, the Mission Ocean, it's a large, complex systems of interconnected projects and activities, and which one of us has a role to play. But the connection of all these parts is all about solutions, concrete solutions for the greatest challenge we have now in our time. So what I expect to hear from you, and I hope the audience will be waiting for the same kind of messages. What are you doing in ClimaRest? What are the solutions? And what could we learn from you in terms of scaling up and replicating and achieving the goals of the Green Deal, and most importantly, to protect 30% of our ocean and strictly protect 10% of our seas. So I don't want to spend much time here on the presentation. I will pass to Marta. She's the project manager of ClimaRest project. And please introduce yourself and please start the webinar.
Yeah. Hello, everyone. Good afternoon. Thanks, Jose, for the introduction. I think we can share the presentation. Yeah, thank you. So my name is Marta Pujol. I'm a research scientist at SINTEF Ocean, which is a research institute based in Trondheim in Norway. I'm in the management team of ClimaRest, but the project leader is Ida Beate Overjorda. She unfortunately couldn't be here today, but I will present the project instead. Next. Yeah. So ClimaRest is an EU-funded research project. It stands for Coastal Climate Resilience and Marine Restoration Tools for the Arctic and Atlantic Basin. It belongs to the mission Restore Our Oceans and Waters by 2030, and it's member of the Arctic Atlantic Lighthouse. The project lasts 3 years. It started in 2022 and it will last until December 2025. And it received funding from the EU with 8.5 million euros. Next. ClimaRest consortium consists of 18 partners. You can see here the name of the partners, and we have represented 7 European countries. Next. And here we can see the workflow of a ClimaRest project. So the project is organized by different work package, which include stakeholder and citizen engagement, marine restoration protocols and technologies, demonstration, large-scale restoration, blueprints, and roadmaps for replication, which also include economic and governance models. And finally, we will develop an online toolbox. And by the end of this project, it is expected to be new projects connected to the replication areas, and it is expected that the online toolbox that we will develop, it continue active and it continue expanding after the lifetime of this project. Next. Yeah, in ClimaRest, we aim to develop restoration tools, and we want to demonstrate these tools in 5 different sites across Europe. So in the north of Europe, we have Svalbard, and then if we go south, we have Ireland, France, Spain, and Madeira. Each of these sites are representative of different ecosystems, but also of different anthropogenic and climate change pressures across Europe. And we will hear more about these demo sites in a few minutes. Next. Yeah, We also have replication areas in CLIMAREST. We aim to extrapolate to other locations the experience from the demo site. So this project incorporates an open call, which is now open until the 8th of September. And what we aim to find 5 comparative areas in different sea bases where we can replicate the experience from the demo sites. You can find more information about this open call in the link that Jose will post on the chat, and all the information about this open call is in the website. And if you have questions related to that, there is also a contact email that you can reach us for any questions related to this. Next, please. Yeah, citizens and stakeholder involvement is a big part of CLIMAREST. It is as well a big part of the mission itself. So we have one of our main goals is to increase ocean literacy in the demo site areas, and we have different activities. For example, campaigns for pollution reduction. We have education programs. We engage with snorkelers and divers for biodiversity registration. And we also aim to develop mobile apps for species registration. So those are some of the activities that we have in the project. Next. And as I was saying, one of the main innovations of this project is the digital toolbox. The digital toolbox is an online decision support tool for marine restoration. It contains specific tools that have been demonstrated in the 5 demo sites in the different ecosystems across Europe. And it uses the EU digital infrastructure such as Emonet and Blue Cloud. And one of the most important parts is that this tool will be open for addition of new tools, protocols, and habitats beyond the lifetime of CLIMAREST. So we aim with this to help to develop best practices for marine restoration. Next. And this toolbox is hosted in the website of the Society for Ecological Restoration. So we have the SER Europe affiliated to CLIMAREST in the consortium. So you will find the toolbox in the Restoration Resource Center of the society. Next. Yeah. And that was the kind of general presentation of CLIMAREST. But now we will go more in detail in each demo site. And for that, we have the demo site leaders or project members that will present each site. So the first site that we will present is Svalbard in Norway, and Anatoly Sinitsyn will present. He's a researcher at SINTEF Industry and he works in the infrastructure department. So Anatoly, the floor is yours.
Thank you very much, Marta. And it's a pleasure to be here today at the Blue Mission webinar. I will present this presentation on behalf of Hanne Kvitsang, who is the site coordinator of the Arctic demo site. We developed this site together as 5 organizations, which are SINTEF, Technical University of Denmark, the Norwegian Institute for Nature Research, Norwegian University of Science and Technology. The site is located on the midway from Norway mainland and to the North Pole. It's about 1,000 kilometers to the both directions. The site is located close to the town of Longyearbyen, which is so-called capital of Svalbard, or the biggest settlement in Svalbard, with population of about 3,000 people. So the site is again is located at the coastal zone of Longyearbyen in Advent Fjord. Next slide, please. Svalbard is a special place. It's one of the places in the globe which experiences the highest rates of climate warming. In some places, or at Svalbard, the climate warming is 5 to 7 times faster than average in the globe. In addition, Svalbard is located on permafrost. That means that only the upper approximately a meter of the ground is unfrozen during summer, and down the soil profile, everything is frozen permanently. Yet the active layer becomes thicker and thicker from year to year due to climate warming. The next slide, please. We have two contexts for the needs for the research we do at Arctic demo site. The first one comes from the increased coastal erosion across the Arctic in both natural conditions and in urban settings. And that is due to increased wave activity and thawing of permafrost. The second need is that in many small Arctic places, the switches released in the sea still untreated. And this creates a danger for benthic communities. So we handle these two elements in two tasks or subtasks in our demo site, which we will look in more details on the next slides. The next slide, Marta, please. And then let's start with coastal erosion. So what we plan to deliver and what we are working at. Firstly, we will develop a framework for integrating Arctic-specific coastal erosion processes in the existing framework of the Coastal Engineering Manual, developed in the US with the last edition that was released about 15 years ago. And then, secondly, we will provide a manual or a review with state-of-the-art of existing coastal protection solutions applicable for Arctic settings, with focus again on urban settlements and urban applications. We will run several comparative computational studies to define sustainability of different solutions. And those points, point 1 and point 2, will pave the way for design of Arctic coastal protection solutions, which would be sustainable and which would enhance biodiversity. Thirdly, we will develop and build a blueprint or a prototype on a demonstration case in Longyearbyen. The next slide, please. This case will be built and it is under development together with the Harbor Administration Longyearbyen. Could we come? Yeah, we come back to the slide. In Longyearbyen, in its central part, we have a coastal stretch of about 400 meters, which is under coastal erosion. You can see a picture of it on the lower left-hand side corner of the screen. And for this case, the prototype will be developed on the principles of co-creation. This includes several elements. Of course, the prototype will be suitable to handle coastal erosion. It will be tailored to enhance biodiversity in the area. Technically, it will include reuse of local materials. We plan to reuse timber from the Svea mines in Svalbard that were decommissioned a year ago. We plan to use local materials, And also we will engage public in discussion in order to find out other needed elements or functions, such as societal functions, cultural functions, probably some sport functions, and etc. And then the size of prototype will be approximately anything from about 10 meters to 20 meters. Perhaps even bigger, but that will be tailored to additional funding from the local stakeholders. And then we go to the next slide, where we look at the second task on the sewage treatment. Here we will perform 3 activities. In Longyearbyen, they just began a coarse treatment of the sediments in the sewage. And together with local municipality, we will develop societal campaigns to engage citizens to reduce the content of large particulate matters in the sewage. And on the picture on the left, you can see like some muddy material, and it's a bit like difficult to imagine how many different things you may find actually disposed in the sewage system. Quite surprisingly, yeah, the picture, yeah, you might find different things there. Then we will increase the knowledge on efficiency of primary and secondary sewage treatment in the Arctic fjords for removal of nano and macro contaminants. And we will increase knowledge on technical, cultural, and organizational barriers for implementing sewage treatment in the Arctic. The next slide, please. Again, you may ask why we are doing it in Longyearbyen. Well, that's the place with the highest observed erosion, highest observed rates of climate change, also with quite high erosion rates in the area. It's a place where we see increasing pressure from human activities, but at the same time, it's like a logistically accessible place, and it's a representative site for many small Arctic communities. The next slide, please. As a replication site, we bear in mind several places in cold regions, and one of them, for example, Greenland. But where the replication will take place, we will see based on the outcome of the call in September. I think that was mostly all from my side.
Yeah, thank you, Anatoly. Now we move to Ireland case. So we have Ricardo Bermejo from the University of Malaga and Liam Morrison from the University of Galway. And I think Ricardo will present the Ireland case.
Okay, thank you, Marta. I'm Ricardo, and I'm the co-leader with Liam of the work package related with the seagrass restoration. Next slide, please. First thing is to say that seagrasses are not seaweeds. The seagrasses just follow a similar evolutive pathway like the dolphins. You know, between 300 and 350 million years ago, some green seaweeds were able to adapt to the land conditions, evolve there, and produce the higher plants, and some of the higher plants just between 700 and 700 million years ago came back to the ocean. These are different to the seaweeds because they can thrive in sedimentary environments, like usually seaweeds are thriving in rocky shores. Also, the biomass of these seagrasses is more difficult to degrade than the one of the seaweeds, just in general terms. Next slide, please. The seagrasses can form large meadows that can be seen from the space with satellites, and they play an important role in ecosystem functioning. and provide different ecosystem services like the blue carbon sequestration and also nutrient sequestration, which in the current context of climate change and eutrophication is important. Also the complexity of the canopy that they form just allow for a high environmental heterogeneity and increase the biodiversity because there is a higher number of subhabitats, and also play an important role in sediment stabilization and coastal erosion. They just change the hydrodynamic and favor the sedimentation, or the leaves can protect the coast. The leaves that are washed out are protecting the coast from erosion, and also the calcareous organisms that are living on the top of the seagrass leaves. When this degrade, just produce sand. This is really important in areas of the Mediterranean where there is a little, well, another tropical areas when there is little loadings of sands from rivers. Next slide, please. In Europe, the conservation of the seagrass has been really bad during the 20th century. One of the most important events, just explaining the degradation of seagrass meadows, was the waste disease that occurred in the 30s, and it was produced for a kind of mold just that decimated most of the population from North America and Europe. It was affecting mostly to Zostera marina, which at that point was one of the most abundant seagrass species. Also, another important pressure was eutrophication following the Second World War, with the generalized use of synthetic fertilizers that allow the development of industrial agriculture, also produced an imbalance in all aquatic ecosystem that affected negatively to seagrasses. In the 80s, with the improvement on water quality conditions, there were not the expected recovery all of these communities like seagrass meadows or kelp forests. Next slide, please. In Atlantic European coastal water, the main seagrass that we have are Zostera anolti, Zostera marina, Cymodocea nodosa, and the species of Rupia. Although the species of Rupia sometimes is controversial because are not considered seagrasses. Next slide. Okay. Also, the conditions in water quality improve. There is still many stressors that can preclude the restoration of these habitats. These pressures are mainly eutrophication, biological invasion, change in hydrodynamic conditions, increase in temperature, enhanced grazing because of the arrival of new glaciers from southern latitude with the global warming, or the mechanical damage by human activities like the trampling, the anchoring, or the trawling for fishing, or scallops, for example. Next slide, please. We started a small a quick scoping review, just looking at the scientific literature where the restoration action has been developed in the field and has been published in peer-reviewed journals. Here we observe that the species that we are going to work on, which is Zostera marina, has been the most studied. In the Atlantic coast of Europe has been also Zostera marina, and in the Mediterranean, Posidonia oceanica. Next slide, please. We can see also an increased interest on seagrass restoration. This is as we can see, for the number of papers published per year during the last 40 years. Also, we did a small meta-analysis of the data. Here in purple, we have the successful experience. It lines the partially successful experience, and in gray the unsuccessful experience. And we can see that for our conditions, maybe the transplantation is going to be more efficient than the use of seeds, which is called here recruitment enhancement. Next slide, please. Also, this is important for the Marine Strategy Framework Directive and for the Water Framework Directive, that there is not a univocal relationship between the water quality and the ecosystem states. For example, we have a lot of shiftings from seagrass communities to macroalgal blooms. And we can see that here we have a kind of C-curve. It's because of in this part, in the upper part, in the seagrass that can be in high nutrient concentration, is because of the resilience of these communities. And in the lower part, it could be for that we can see the macroalgal blooms. It could be because of the negative trace-off. The new macroalgal bloom is precluding the recolonization of the seagrasses, or because the seagrasses are taking too much time to recover the areas where they were previously. Also in Ireland, we has been observed a lot of shift from seagrass dominated communities in estuaries to macroalgal blooms, mainly because of the agriculture and wastewaters. Next slide, please. The two sites that we are going to study are in Galway Bay, the Rushing Bay, which is a small bay in the area of Galway, where there were some seagrass meadows in the year 2000, but just disappeared because of the urban wastewaters. This urban wastewater has been diverted to a wastewater treatment plant, and we will try to boost the recolonization of this area by transplantation. In the case of the Trolley Bay, which is in the south, the trawling for scallop fishing is also produced the degradation of some of the seagrass meadows, and we will do also some activities just to restore these areas. Next slide, please. Here we can see an overall view of the Trolley Bay, and the area that we are going to restore, or we are trying to restore, is the one in the south of the bay. Next slide, please. We can see here also the area of the Rushing Bay, where we will perform this restoration in collaboration with the Galway Atlanta Aquarium. But this restoration is going to be most focused on citizen science and science dissemination. Next slide, please. Also in southern Spain, we are going to develop some explorative activities of restoration in the Paraje Natural de Mar Cerro Gordo. Here there were large meadows until the year 2005, when suddenly all these meadows just disappeared. Now they are considered extinct, but during the last weeks, one of our student, Africa, just found some leaves washed out of Zostera marina. Then we have hope that there is still some stands, some remaining stand of these species. Next, please. Next, please. Next, please. Here we can see the all distribution of the seagrass meadows in the area. Next. Also, we are trying to engage as much people as possible, and one of the stakeholders who is working really close with us and is doing an amazing work is Sheldon, is doing the mapping of all the seagrass meadows in Trolley Bay, and also the monitoring of the growth, and as well the phenology, which is going to provide really important information for the collection of seeds. In our case, we are going to replicate the work that Ed has been doing in Trolley Bay in the Connemara National Park. Next, please. Finally, here we provide a list of stakeholders that have been actively participating in this project, and we hope to increase this list as soon as possible. Thanks for all, and I gave the floor to the next speaker. Thank you.
Yeah, thank you, Ricardo. Now we move to France, the demo site in France. So Martin Perrault will present this case. He's the CEO from Seaboost. So I give the word to you.
Thank you, Marta. and hello everyone. So I will just present the demo site, the French demo site focused on native European oyster reef restoration. Next, please. So we decided to focus on the native European oyster as once was a keystone species that formed critical biogenic habitats along Europe's coastlines. It's gradually degraded due to multiple pressures, including overfishing and more recently in the species history, parasites. And today is in a critical state of conservation. However, there are a few remaining populations throughout Europe's coastlines, and for the past decade, there have been a growing number of initiatives to restore the species in various forms. In France, several projects have already been implemented to restore the species, and these projects have focused on restoring the species in the form of a cohesive biogenic reef formations. These initiatives have been led at the meter square scale, several meter square scale, and there are still many challenges left to be able to upscale this restoration. Now, we decided to focus our effort on this species as it's known and described, and oyster reefs are described to be globally interesting for many ecosystem services, such as water purification, habitat for many species, including fishing resources, shoreline stabilization, denitrification, etc. And so, and the objective of this call for resilient ecosystem, for resilient coastal communities, we decided to focus on upscaling restoration for this specific species. Next, please. Our objectives in this project are to, first of all, continue developing and validating operational techniques that can be deployed on the field for large-scale native oyster restoration. As I mentioned, past initiatives for reformation of biogenic reefs have occurred at the meter square and scale, and our objective in this project is to work at the scale of 100 to 1,000 square meters. This is one first very important objective of this project. The second objective is to improve knowledge and characterization of the ecosystem services that this habitat can supply. And I'll develop a little more later on in the presentation which specific ecosystem services we'll be focusing on. And finally, of course, and this is an important objective of the CLIMAREST project in general, is to see how we can engage local stakeholders and come up with new types of project development and funding models to take these restoration projects to the next level in terms of upscaling and multiplying also the initiatives, restoration initiatives along the European coastlines. Next, please. So here are just a couple of pictures of the species, and on the left you can see what remaining oyster beds can look like in a couple of sites that we will be working on in the south and in the western part of France, in Brittany. So the species is sort of scarce from a couple of individuals to more than 20 individuals per square meter. The species is present as in the form of individual oysters or small clumps, and our objective is to restore the species in the form that you can see on the right-hand picture, which is the result of a past initiative, successful past initiatives of restoring the species in its form of a cohesive reef. So this is what we're targeting in terms of habitat restoration. Next, please. We'll be working mainly on 2 demo sites within the CLIMAREST project, both situated in Western France, in Brittany, in the Bay of Brest and the Bay of Quiberon, which are 2 contrasted sites, both in terms of environmental conditions, of also human influence, where we have quite an urbanized coastline in the Bay of Brest and more open coastline, but marked with quite intensive shellfish farming in Quiberon. The species and the surrounding natural habitats are also very different in both sites, and this will bring different types of restrictions in the protocols that we can or cannot deploy for species restoration. Next, please. So today we're a little more than 6 months into the project, and we have deployed recently at the end of June the first series of restoration substrates. So in the Bay of Quiberon, our logic is to deploy substrates on which the oyster larvae can settle and then grow and form a reef over time. The idea is that these substrates act as a form of a skeleton or a frame for development of the reef, and that they stay in the water and may degrade over time, leaving the area with restored reefs. So we deployed 33 structures in the end of June, covering about 1,000 square meters. We had deployed them at the peak of the larvae concentration in the area. There are in this area there are of course remaining oyster beds. It's known to have good recruitment, and so hopefully in September we'll be able to confirm that we have had good settlements of oyster larvae on our structures, initiating the restoration process. Next, please. Further on into the project, we will be carrying various monitoring and modeling work to, as I mentioned earlier on, be able to qualify and quantify different types of ecosystem services. So we will be focusing on the water filtration capacity, and this work will be mainly done in the Bay of Brest, around the previous restored reefs have been restored in frame of past projects. We will be working on carbon capture in both sites, so trying to improve knowledge on the carbon sequestration capacity of such restored habitat. We will be modeling also the effect that restored reefs in the Bay of Quiberon could have on wave action and coastal erosion mitigation. And of course, in that area, a sustainable supply of oyster spat is something very important to maintain the oyster farming industry. And so we will also be monitoring this. We will be monitoring in a more general way the biodiversity that can develop around restored habitat and the scale at which we're working with in this project. And of course, we'll be monitoring the general performance that our protocols enable for reef reformation. I will stop there for today for this presentation, and I'm open to any follow-up interactions that you would like to have for more detail on this project. Thank you.
Thank you, Marta. Now we will move to the Spanish demo site, and Pablo, he's a professor from the University of Alicante. He will present this site.
Hello everyone. Okay, thanks for organizing this nice workshop. Please, next. Okay, we are already working at the north of Spain in Galicia, Ria de Vigo, where is, as you know, an area with a very high primary production because it's the upwelling of the Canary current working there. That is reflection in the very high productivity of the area. For example, the mussel production in 2022 was, yeah, you can see 220,000 tons. I mean, we have, it's very relevant aquaculture in this area. Maybe you don't know how is the RAF, is no long lines, specific kind of production where you have all these thousands of production system of mussels with these lines, vertical lines, that is, I mean, is affecting in some ways to the ecosystem. Next. Okay, the relevance is that this area with a very high primary production due to the upwelling, of course, is affected by climate change, and is also affected for several human activities. Marine aquaculture, as we see with the mussel production, fisheries, transport, pollution, tourism. But all this primary production is offering a lot of ecosystem service, as well the aquaculture and the fisheries. And in general, this soft sediment, that is one of the most important habitats in the coastal marine environment, is very relevant from the point of view of the global carbon cycle. I mean, the organic matter in the sediment is quite relevant for the climate change adaptation, with the possibility of a burial, you know, or removal of the carbon that is sediment. I mean, this habitat that is very important from the point of view of the ecosystem service is also affected for many activities, and under our umbrella of the climate change is having a strong change, you know, and modifying. Next, please. The actions, what are we doing? We are trying to understand what is happening because it's a quite complex area, you know. We are modeling the ecosystem functioning in terms of carbon, nutrients input under this scenario of the climate change, considering mussel production as the main driving stressor and the other anthropogenic impact. Okay, we were following different kind of activities to do that, you know, using geographical system, systems to analyze at the scale of the RIA, how are the different ecosystem service changing depending of the climate change and depending of the other anthropogenic stressor. I show you an example that have we already done for the College of University of Vigo, that we are working together, University Alicante of University of Vigo, that is similar to what is the natural capital of the seagrass, and we will try to identify what is the natural capital and what is the ecosystem service of this soft sediment without seagrasses, but that are affecting for mussel production. Next, please. Okay, what are we doing? We are doing different things for understanding how it's affecting in different ways the mussel production system, and how can we do some actions to increase the quality of the habitat and increase what is the ecosystem service. We develop and implement an artificial structure that are already underwater in habitat affected by mussel production, and you know, in areas that is controlled. We just sand bottom, no, without the mussel production. we are doing in different sites and with control and doing a monitoring of. Next, please. Doing the monitoring of how the Necton community, the benthic, we can see that already in few weeks, there was full of squid eggs, and echinoderm and fish are already colonizing. Next. And we are doing also experiments of the respiration of this system. Why? Because we think that we can reducing the impact of the aquaculture mussel production system if we can increase the community that is using this organic matter and increasing the secondary production. We evaluate, you see these plates, they are already prepared for take it out and take to the lab and make this experimental respiration after months that they will be colonized for the fauna. Next. Okay. Another implementation is restocking and working with a specific flat ship species, like the European lobster or Maruga marus, in this system affected by mussel raft, and they are already with a lot of fishing pressure. Okay, for that we must know how they can be using this artificial habitat. And if you see there is a marine protected area, the TIAS, that is at the left side of the screen, and all this mussel production that you see that is inside of the RIA. And next, we will be doing, sorry, can you go back, Jose? Sorry, I will try. We will be doing telemetry, with all this tagging system that is in black, acoustic telemetry, to evaluate how lobsters that are from from fisheries natural one and lobsters that come from aquaculture, how they are using natural habitats in the marine protected area of TS and the habitats that we built artificially under the mussel raft and in another control area with sun, to understand if they are optimal habitats for them. Next. Another implementation action is restocking, to develop a restocking protocol, but with some interesting technological approach for reducing the mortality, using the juveniles, for restocking, and to see if with this new technology we are able to reduce the mortality and increase the population in the area. Next one. The results, what we want to know and to develop actions, for adaptation and restoration related to the climate change and multi-stressor in this very complex area of upwelling at the north of the Canary Current in the Galicia Rias, increase their resilience for, making the recruit of the species and the biodiversity higher with these new habitats, enhance the endangered species as European lobster, and that can be like can be developed for another target species for fisheries. Develop an adaptive management of the resources, knowing very well which are the ecological service and which are the impacts that are affecting them. And of course, and it's what Marta explained, CLIMAREST is highly focuses in the society implication on marine conservation and climate change through citizen science, and the implication of the stakeholders. Next. And that is, thank you very much.
Thank you, Pablo. So now we move to the last demo site that we present today, and it's Madeira. Paola Parretti, she's a researcher at Mare, and she will present the Madeira case. I give the word to Paola.
So hi everybody, and thank you to organizing this, and thanks to the ones that are still with us and hearing us. So next slide, please. So what I'm going to present you today, it's a sort of history. So how we identify our threats and how we are going to do it to start to restore our ecosystem. So here at Mare Madeira, we are at the Ecological Research Center. So of course, we conduct a lot of underwater monitoring. Next slide, please. So what we do usually is we collect information about the abiotic characteristic of our ecosystem and of course the biotic characteristics. So usually we do a visual census to assess the population of a fish, of a macroinvertebrate, and of our sessile community. Why we do this? Because we want, of course, to characterize the typical assemblages of our coast, and we want to determine a possible correlation between the diversity of, for instance, the fishes, the macroinvertebrate, or the sessile environment. And of course, in this way, we have a huge baseline for monitoring. Next slide, please. So here is just to give you an idea of what we can do with our data. So what you can see here is a map of a Madeira island, and the small dots represent the fish community in our monitoring site. As you can see, we usually monitoring on the south coast of the island because it's the one that is most accessible, while the north coast is pretty rough, so we cannot go there every year. The next two slides actually are quite the same, but just to give you an idea, you know, what we have with the macroinvertebrate and with the sessile community. So what's happened when we collect all this data? You can go to the next slide, please. And to the next also. Okay, thank you very much. So here, when we combine the data of, for instance, the community of our macroalgae with our invertebrate, we discover two really clear patches. So we have on the blue circle, basically is all the structure of the benthos dominated by sea urchin, while on the left side, so the green circle, is the community that is macroalgae dominated. So here we discover a clear pattern, and among the sea urchin, we have predominantly three species. So we have the Diadema africanum, the Herbacea, and the Spircinus. And we see that when there is a huge amount of the Diadema africanum, we have definitely really low percentage of macroalgae cover. So then what's happened during our monitoring program is that we discover in 2018, you can go to the next slide, please, a huge mortality of Diadema. So as you can see here on the left picture, you have an healthy Diadema, and on the right side you have a sick, well, in this case is almost dead Diadema africanum. So we notice this huge mass mortality, and we ask, next slide please, to our citizens to help us in monitoring this huge mortality. So we develop an online survey addressed to basically everyone, diver, fishermen, and spearfisher, to help us collect this information about the sea urchin mortality. And as you can see, the bigger the dot, the bigger is the mortality. So we have the mortality basically in all the south coast of the island, but please pay attention that on the north, basically no one is going there, it's almost inaccessible. So next slide, please. So what we did after, we said, okay, let's observe our community in 2017, which were before the mass mortality event, then in 2018, which is during the mortality event, and then the year after. So when the sea urchin are starting, they are not present, they are starting coming back, but not in a huge abundance. And what we saw in our macroalgae community is that we pass from a barren state. So a barren state is where you have the seafloor completely white, so no algae at all, just sea urchin, to a macroalgae dominated state. So clearly we identify that the abundance of a sea urchin was our, let's say, danger factor. So next slide, please. So, and we were able to discover a threshold. So it's like when the population of a sea urchin is a danger for our macroalgae and when is not. So we see that when we have a 50 sea urchin per 100 square meter is where definitely we starting with some issue. So next slide. Another thing is that we ask, so how was the population of the macroalgae in Madeira a few decades ago? And as you can see in this picture, so you have always on the left side the oldest situation, so 1995, and then you have more or less the present day. So in Madeira, we used to have a macroalgae. It's not something that we just want to have. There was. But unfortunately, we have this huge abundance of sea urchin that transformed the situation in a complete desert. So next slide, please. So what we want to do in the CLIMAREST. So we want to work in two different directions. In first direction, we want to, let's call, do a forestation effort. So we want to have more macroalgae on our sea bottom. And then we need to do the graze control. So next slide, please. How we are going to do our forestation effort? So we are trying different techniques. The first is just to collect the macroalgae from their natural habitat and let them grow in the lab and then try to retransplant. And the other technique will be already put this micro plate in the field, let them colonize naturally by our macroalgae, and then transplant this plate into the field. And as you can see, we are already starting doing this and we are having some success for the moment. And the last option will be having artificial reef to be colonized. So we deployed this artificial reef under the water and we will let them colonize. So next slide, please. For what concern the grazing control, we still have no select the most efficient way. So we are still evaluating if we're going to do some sort of caging, so to exclude the sea urchin, if we're going to remove the sea urchin, or if we are going to find something else. Next slide, please. So where we are going to do this? So in Madeira, we have actually several number of marine protected area, but we are going to work in the most oldest one. So is the reserve of Garajao, and you can see it here in the green color. So we select 2 hours inside our marine protected area and 2 hours outside our marine protected area. And we are going to work in both ways. So on one way, we are going to reduce the pressure of a sea urchin, and on the other way, as I told you, we're going to increase the algae cover. Next slide, please. Here you have just a quick example of our experimental design. It's basically what I told you before. And of course, next slide, please. We are not able to control all this area alone, so we ask the collaboration of different stakeholders, and in particular, we will work with diving center and divers. So as you can see here in this map, we have several diving centers located all around Madeira Island. And in particular, we have several that are inside the marine protected area in which we will be working. So we are already asked this diving center if we can deploy this different structure in their house reef, so where they usually go to dive, and they are super enthusiastic to participate with us. And another important aspect in which we will collaborate with the stakeholder is to help us in monitoring. And how we are going to do this? Next slide, please. So we develop an app, which is the Dive Report app, which is already available. And we will provide this app to our diving center in two ways. So one, they can use a tablet and just download the app. And in another way is we develop this amazing kiosk with the seahorses shape that will be left in the diving center. And in this app, we made, next slide, please. We select several species that are indices of our biodiversity status and good health status on the environment. And we ask to the diving center after each dive to select which species they saw. And after select the species, they can also select the abundance of each species. And we already conduct a study in which we compare the data obtained with this app and the data obtained with our monitoring survey. And we can say that the results are really, really nice because the data are really comparable and they ended up with the same result. So let's say that we prove that citizen science work, and is a great help for science. And with this, I thank you very much, and I finish.
Oh, thank you so much, everybody, for these very interesting presentations. So, we are a bit after the hour. So, but it's still time for 2 or 3 questions. I've seen a lot of, questions and answers here on the Q&A box, but people from YouTube and also for the recording won't be able to see what was discussed. So I will go back. I'll take 2 questions only for us I'll go back to Rachel Cade. Hello, Rachel. Nice to see you here, and I hope to see you in Iceland very soon. So you asked about the permits. It's interesting to know for all these very interesting projects. They should like to know how difficult is the licensing process to carry out the interventions. So maybe Pablo and Anatoly, can you repeat your answers and also, reply to Rachel's second question is related to the same about the permits. So just to be on record.
Hello, I can be very quick. It's that depending of the politics and the administration, how they open are to do this kind of thing. You should be lucky. What is very good thing is to have a risk assessment of your activity and show them to say, okay, we are doing that. That could be bad. That could be good. That will be the solutions, and that will be, the problems that we can produce to other stakeholder or user of the area, and explain and show the risk assessment of your activity, and you can get easily a license if it's possible for the depending of the regional or national administration. That was my recommendation.
Yeah. Any further comments, Anatoly?
And for our part, we yet have to initiate the process with the permits, but we had initial consulting already with authorities, and we found out that it's much easier to get permits for the urban environments compared to, let's say, a wild coast or some like natural reserves. We had an idea to build our prototype in more isolated settings outside of the town. But then we find out that the minimal time to get permit is 1 year in Svalbard. It's very strict when it comes to nature reserves and environmental protection law. So that's inside of town. It's more. It is controlled by the local government, and then usually we were informed it's a matter of few months. That's how it works in our case.
Thank you so much, Anatoly. So I have a question from David White. By the way, David will be presenting Prep for Blue in one of upcoming sessions. And so David White says that, great to hear of the really interesting demo sites. Question for Marta. It will be a challenge to present results to the Commission with the aim of successful demos being upscaled in phase 2 of the mission in good time. How do you think we can best work with the commissions to make sure the results of phase 1 are available to influence later call topics in good time? It's a very good question.
Yeah, that's a very good question. Interesting and difficult to answer. But I think that it's important that we kind of analyze our results, and we also work together with the other projects in the Lighthouse, right? And then we try to work together to define which are the standards, or which are the topics that should be included in the next phase. So I think we have to be able to kind of prioritize according to our experience, and also having close contact with the project officer and the commission, and try to find the right way to communicate that for the next phase. Yeah, but I think there's something that we need to discuss all together within the project, but also outside the project in the Lighthouse. And we try to work together to find the right way to communicate our results. Do you agree, Jose?
Oh, yes, definitely. It's something that we have to discuss with all the other projects already approved in Lighthouse, and also together with the other Lighthouse, because they might have kind of solutions related to restoration. And as you've seen, we have solutions related to marine pollution, for example.
Yeah.
So,
Hopefully there are some events that we can be together and work together to find the right solutions.
Yeah, I have to say that the projects of the Atlantic and Arctic Lighthouse are working very closely together. We as a Blue Mission, we don't have any case studies. We are using the Svalbard case and for our case study follow-up. So I was supposed to close now, but I have to take this question because it's from Natalia, my colleague from the center. If I don't take this question, she will kill me. So congratulations to the speakers for the nice overview of the project. And Natalia's question is for Pablo. Regarding ecosystem services on which CLIMAREST is focusing, are you considering cultural or recreational services, for example, in the CS Island area?
Yes, we are considering all of them, but we should evaluate which are more important. We are trying to use Invest system software, okay, that is already developed for doing a kind of geographical information system, have modeling that under climate change. That is the main thing that we are doing, okay? Is modeling ecosystem service, but modeling for in a short period of time what will happen. We know with summer where it's happening now in Spain. We are getting 40 degrees, okay? The answer is yes, and we evaluate, and it's part of our colleagues. It's here, Laura Leiva and Paula Davanda, they are the PhD student working on that, and that will have soon part of the results. Thank you.
Yeah, so again, one last question, sorry about that, but it's from Belinda from MPA Europe. I mean, it's one of the projects that was just approved, so they are very interesting on your solutions. So the final question is there an urchin predator missing from the ecosystem in Madeira that could be reintroduced to regulate the urchin population? That's for you, Paola.
Yes. So as far as we know, no, there are not predator missing, and if they were, we are not really keen of introducing new species in the island, because we are already with a lot of non-indigenous species here. So I don't think that this could be an option, let's say this.
Okay, so I have to thank you all for your fantastic presentations, and so for the attendees for the very interesting questions. And we'll be back next week with the Remedies projects, which is focused on marine pollution. So again, thank you all, and good luck, ClimaRest. We hope to use your solutions to solve, I mean, the challenge that we have ahead. Thank you so much.
Thank you.
Bye-bye.
Thank you, everyone.
Bye.
Bye-bye.
Bye.
Bye, everyone. Thank you.